that's used after DNA replication, the so-called late promoter, and the exact one varies somewhat depending upon the strain of HPV, but with that alternative late promoter, then there's a different polyA site that's used on the late transcript that then is spliced to allow us to encode our late proteins. And this is figure 1 of your handout that shows the genomic organization in a circular schematic, because our DNA genome is a double-stranded superhelical DNA molecule. But in many texts and presentations, the genome is shown in an open or linear format, and that's why I gave you a figure 4 so you can see it both ways. But again, it depicts our early promoter and early polydenylation site, and a different promoter and a different polydenylation site that's used late. All right, so then among our genes that we express, we first express E1, and so E1 is going to be important in the replication of DNA. It's conceptually much like T antigen in that the early E1 protein that is expressed is important to give the initiation of our HPV DNA replication precision. It's the binds to the ORI and allows for the DNA replication. Then we have E2 that's expressed, and so E2 acts to regulate transcription. So E2 then functions with the cellular RNA polymerase and general transcription factors in regulation of transcription, but also functions in DNA replication together as well. So E2 is in 2 positions on your map. And then E3 and E8, as far as I'm aware, the functions are still a bit elusive in terms of precisely what these proteins are doing. But then E4 was elusive, but we know where E4 is doing. So E4 we're going to return to. It affects the cytoskeleton. E5, E6, and E7 play roles in oncogenesis. And so E5 E5 works to alter membrane function. E5 is expressed and is targeted to the plasma membrane of the infected epithelial cell. And E5 seems to play a role in activation of the epidermal growth factor receptor, EGF receptor. And then E6 and E7 are the oncogenes of the virus. And the capacity for oncogenesis mediated by the E6 and E7 protein products differs dependent upon the serotype of the virus. And you'll see why mechanistically in a few minutes. So E6 and E7 are the oncogenes, so they encode the oncoproteins. They're the gene products that's responsible for oncogenesis and ultimately surgical carcinoma. And so how do E6 and E7 work? They work by inactivation of the cellular tumor suppressor proteins, and there are 2 of them that will be inactivated. Okay, so E6 and E7 then will be expressed, and they are targeted back to the nucleus, and within the nucleus they're going to inactivate P53 and RB to mediate the biology. All right, so then, in addition to the early gene products, then we have three proteins, L1, L2, and E4, even though it's an E, it's expressed late, that are late gene products. And so, what do these late gene products do? So L1 and L2 encode the capsid proteins. L1 is the major capsid protein, L2 is the minor capsid protein. And so L1 and L2 are expressed from differentially spliced RNAs that are made from the late promoter, the primary transcript, or spliced in a cap, which, by the way, in the process of processing RNAs in an animal cell, which SB40 was the prototype that led to many of the discoveries. So in our processing of RNA, we have the 5' capping, about the 3' polyadenylation, and then we have splicing. And in general, the temporal order to which these events happen to our RNA, and it will be true for HPV RNAs as well, the 5' capping, 3' polyadenylation, this typically happens quickly. The capping, very rapidly. Capping happens way before the full transcript is actually transcribed by the cell call to machinery. Very early on, the capping happens. Then we get our full transcript, and in this case it's the HPV RNA, the primary RNA, and we'll have polyadenylation, as we had with SB40, or the hexanucleotide signal, standard mechanism. Those modifications protect the RNA because the splicing that then occurs is a much slower process. And so splicing, so the capping is first, polyadenylation typically second, and then splicing is temporally, in terms of the kinetics, much slower. But does occur, and here we're getting two different differentially spliced RNAs that will encode L1 and L2. These capsid proteins then are targeted back to the nucleus. And as late gene products, that expression has occurred after we've replicated our DNA. In addition, the E4 protein is expressed late. And the E4 protein that's expressed is targeted to the cell cytoskeleton. So E4 goes to the cytoskeleton, mediates cytoskeletal changes in the infected cell, and alters the cytokeratins within the keratinocytes of our infected animal. Okay, so that's the temporal regulation in the context of the virus-host interaction. How about the spatial regulation in the context of differentiation state of the epithelial cells? And so in addition to temporal regulation, which is what we've been discussing, we have spatial regulation, which relates to the differentiation state of the infected cell. And so the early genes are expressed in infected basal cells. And so these have not differentiated. And so you first have, in terms of the temporal early late business, first expression of E5, E6, and E7. And when these genes, E5, E6, and E7 are expressed, and we're going to return to E6 and E7 in a bit, but when we have expression of those gene products, that then leads to induction of E1 and E2, expression goes up, and then that leads to induction of DNA replication that occurs of the virus, which is happening in the keratinocytes, which are the dividing basal cells. This then leads to late gene expression in the context of the temporal business of the virus-host interaction, which is the expression of our capsid proteins L1, L2, and then E4 that's altering the cytoskeleton. So it's within then the differentiated cells that we have late gene expression that's robust. And so when we have in those differentiated cells then our late gene expression, we have had viral DNA replication. So DNA replication has occurred. We've built up large pools of the viral DNA. We've made our late proteins. So then we have within the nucleus of the infected cell the assembly of our progeny HPV virions. So HPV assembly of the produced viral particles happens within the nucleus. And then we have an aggression out and release of the progeny virions from the epithelial cells. All right. So back to E6 and E7. So how are E6 and E7 functioning then to mediate the change in growth control of the epithelial cell and differentiation status? So E6 and E7 work by inactivating cellular protein targets. And so they inactivate the two key cellular proteins that are tumor suppressor proteins. So the tumor suppressor proteins are cellular proteins, and these cellular proteins function normally to block cell cycle progression. So they're the brakes. They put the brakes on the progression of the cells through the cell cycle, and as a consequence, you get when they're functioning, you have an inhibition of cell proliferation, so you get fewer cells dividing, and you can have an induction of apoptosis. So an induction of cell death, effectively removing damaged cells. So if you inactivate a tumor suppressor, then you don't have the impairment of the cell cycle, and you don't have the induction of apoptosis, so you have uncontrolled growth of the cell, which is characteristic of a transformed phenotype. Okay, so P53 is one of these cellular tumor suppressor proteins that we touched upon with SV40. And in a normal cell, typically you'd have very low levels of p53, and you get an induction of p53 by viral infection with some kinds of viruses, or DNA damage of the cell can lead to elevated levels of p53. So you end up with sort of high levels of p53. So then, how does p53 normally activate? And this may be review for some of you. So how p53 normally activates, acts, is it activates cell transcription of some key cellular genes. So p53 is essentially a transcriptional regulator. And among the cellular genes that it activates the transcription of is something called p21. So p21 is induced by p53, because p53 is a transcriptional regulator. And so what p21 does, you induce the RNA, and then you make the protein, and this causes growth arrest at G1 in the cell cycle. So if you can make a bunch of p21, then you arrest the cell cycle at G1. Another key gene that I want you to know about is BAX. So BAX is also induced by p53. And BAX then encodes a protein that's a pro-apoptotic factor. So the BAX gene product enhances apoptosis. It enhances the cell death and removes cells. Okay, so that's what p53 is doing under normal conditions. So now we have an HPV dependent upon the strain, and I'll get to the strain bit in a minute. So HPV infects the cell dependent upon the differentiation state of our epithelial cell. We get more or less E6 and E7 expressed, but the E6 is targeted to the nucleus, and E6 binds p53. And high-risk strains of HPV that are highly associated with oncogenesis and cervical carcinoma are very effective binders of p53, whereas low-risk strains, that interaction is very inefficient. All right, so when E6 binds p53, p53 is inactivated, and actually p53 gets modified by a modification called ubiquination, and this leads to degradation. So the formation of our oncoprotein of the virus interacting with a cellular tumor suppressor protein inactivates the tumor suppressor protein, but not only does it inactivate it, it leads to the degradation and destruction of the tumor suppressor protein, so then you no longer have the cell growth control and you have uncontrolled growth. So that's how E6 works. And E6 from, as I said, highly oncogenic strains of HPV interact and mediate this inactivation of p53 most efficiently. How about E7? So E7 is going to inactivate a different tumor suppressor protein called RB. RB is also a cellular protein. RB was discovered in retinoblastomas. Retinoblastomas. It was discovered as being a mutated protein gene product in this type of cancer. And then it was shown ultimately to be a family of proteins, and the key members are P107 and P130. So how does, in a normal situation, how does RB, and that's where the name comes from retinoblastoma, how does RB normally function in the cell? So RB normally functions by interacting with a cellular transcription factor that's important in cell cycle control, E2F. So RB is a cellular protein, E2F is a cellular protein, E2F is a transcription factor. So RB is a negative regulator of E2F. And so there's a cyclin-dependent kinase, a CDK. CDK is cyclin-dependent kinase, so it's an enzyme that's regulated by cyclin. It's a kinase that causes phosphorylation. And CDK then, when activated, causes the phosphorylation of RB. And so when RB is phosphorylated, it dissociates from E2F. So when RB is phosphorylated, then you get E2F that's dissociated, and E2F now is active. When E2F is bound to RB, it's inactive. It can't function as a transcription factor. But when you have a mediation of the activation of the kinase that phosphorylates RB, then that frees up E2F that functions as a transcription factor in the animal cell. And so when this is active, then you have cell cycle progression. So now in the HPV infected cell, we make E7. E7 is targeted to the nucleus. E7 interacts with RB. So when E7 interacts with RB, it inactivates RB. And so RB then cannot interact with E2F to turn off E2F. And so consequently, you have E2F that's free, that's active, so it's mediating this cell cycle progression. So that's how the oncoprotein E7 of the highly oncogenic strains of HIV works by inactivating RB. So this concept of a virus making an oncoprotein, an oncogene product that's causing transformation for HPV and also for SV40, where much of this with P53 was first discovered. These viral oncogenes are working by inactivating the off protein of the cell, the tumor suppressive protein. When we come to retroviruses in the weaker cell, you'll see a different situation with those viruses. The oncogenes actually are active. They're doing something in a different way other than inactivating tumor suppressors. But SV40 T antigen, I did introduce you to the fact that SV40 T antigen binds P53, and T antigen is a large tumor antigen that causes oncogenesis in those rodent cells by inactivating P53. It turns out that T antigen also binds RB and inactivates RB. And so we have with SV40, large T is inactivating both of those tumor suppressors. Whereas with HPV, we have a different gene product, target being the individual tumor suppressors. Okay. So let's move from the cell and the mechanism to a bit of epidemiology and disease. So, in terms of HPV epidemiology, with regard to transmission, transmission is via sexual intercourse. It's the most commonly transmitted sexually transmitted viral infection. And so the estimated lifetime risk, the estimates from the epidemiologists are that in the order of 80% of us have been infected with HPV. So it's pretty high. In the vast majority of these infections, it's asymptomatic. There's no ill effects, the infection's resolved. But that's not always the case. And HPV infections are the cause of greater than 99% of cervical carcinomas. And in the US, greater than 70% of the cervical carcinomas are caused by HPV 16 and HPV 18. So these are high risk. The oncoproteins of 16 and 18 are very efficient in inactivating RB and P53. Also, greater than 90% of the genital warts that are seen in the US are caused by HPV 6 and HPV 11. So these strains, even though they are a major cause of the genital warts, these are low risk for oncogenesis. Rarely seen with cervical carcinoma. Some high-risk HPV strains now are being seen with cancers other than cervical carcinoma. And a couple of years ago, I had Dr. Cox, who was head of the women's clinic and involved in some of the clinical trials of the vaccine, give a guest lecture, and he pointed out that among those other cancers, anal cancers and oral airway pathway, some of the tumors are now showing up as associated with the high-risk HPV strains. So what's the progression? What have we learned about progression of the oncogenic infection that leads to cancer through inactivation of the tumor suppressors? So in terms of the epidemiology, the infection usually happens of young women, late teens, early 20s. And this then leads to intraepithelial lesions that might be seen in the late 20s. And then it's like several years later that invasive cervical cancer presents, and the mean age is about 50, so a substantial period of time. There's a classification that's come to be known as CIN's classification, and histologic classification The acronym stands for cervical intraepithelial neoplasia. And there's 3 rankings. Let's see, P53, we had that. We get a P53 controls P21 expression, alters the cell cycle at G1, alters apoptosis. Yeah, so the cervical epithelial neoplasia, CIN, dysplasia classification, and how that relates to infection. So there's 3 scores, 1, 2, 3. 1, mild dysplasia, and you have this as figure 7 on your handout. Mild dysplasia. And in terms of the virus, this is where we see a productive infection. The virus is replicating, and so the viral DNA is not integrated. So it's a productive infection, we get virus replication, the viral DNA not integrated, it's episomal. So what we mean by episomal is that it's free as our circular molecule, it's not integrated. SIN2 is moderate dysplasia. And then SIN3 scores severe dysplasia. And this then would be carcinoma. And in this case then, we're not producing virus. The viral DNA is integrated into the chromatin. And as a result of that integration, the expression of E6 and E7 is elevated. And that's summarized on figure 7 of your handout. SIN1, virus is replicating, it's active infection. But by the time we get to the carcinoma, SIN3, we're not producing virus, the DNA has been integrated, and the expression E6 and E7, the oncoproteins, is elevated. And at that stage, then, we don't have the circular episome free in the cell, but the DNA of the virus has been integrated in a way that then leads to the elevated expression of E6 and 7. Okay, so many years of basic research led to the understanding of how HPV is replicating and interacting with the host to alter the changes. And there was one investigator in Germany, Sirhausen, that was really the pionear. And Sirhausen received the Nobel Prize in 2008 for really characterizing HPV and how the virus replicates. He recognized that there were different strains that have associated then with them different biologies, including the cause of cervical carcinoma. And so this then led to a vaccine. And so that was a late-breaking development for the class a couple of years ago, actually nearly 10 years ago now. The vaccine that was approved by the FDA, Gardasil. So what is Gardasil? The so-called cervical cancer vaccine, of which some of you in the room hopefully have been immunized with Gardasil. So you have this as figure 8 of your handout. Gardasil is what we call a quadrivalent vaccine. and from our discussion actually of influenza, you would know what quadrivalent is. It's actually directed against 4 different viruses, quadra. So the vaccine is active. What 4 strains would you pick from what I told you so far? You pick the 2 that are responsible for the the bulk of the cervical carcinoma, and the 2 that are responsible for the bulk of the genital warts cases, and that's the 4 strains. So it's active against HPV 16 and 18, so that's about 70% of the carcinomas. And it's active against 6 and 11. There may be a modest cross-reactivity against others, but these are the main targets, and that's greater than 90% of the genital warts. So it's quadrivalent, and it's also what we call recombinant. It's a recombinant subunit vaccine. So what that means is we're only expressing a part of the virus, and the part of the virus happens to be the L1 gene. So the L1 gene that makes the L1 protein, the L1 protein is expressed as a recombinant protein. It's actually expressed in yeast and purified from yeast. And that's done for these 4 different serotypes of HPV. And when you take purified L1 protein, it's a concept that we've already talked about, self-assembly of virus particles. So L1 will self-assemble to virus-like particles. So L1 self-assembles, interacts to give these particles that look like a virus. They're called then VLPs. And so it's the VLPs that you've been immunized with. And so then what happens? So when you're immunized, you generate antibody against L1, and that antibody then neutralizes the virus by impairing the interaction with the receptor. And so that's the essence of how Gardasil then prevents infection. So prevents HPV infection. So there's high promise that over time, if one can target, the key serotypes of the virus, one has a chance of eradicating cervical carcinoma. There are some social issues, of course, in that immunization has to happen at an early enough age before the infection. And so that's one aspect that I think is, to some extent, is being dealt with. The other of which is now that, of course, men could and should be immunized as well because they're a vector for transmission of the virus. All right. So I'm going to change gears now. We're going to leave the small DNA circular viruses, Papilloma and Papillomaviruses, and move to our next virus family. And that will be the Reoviridae. Any questions on papillomaviruses before we leave this topic? Okay, so the next family then that we're going to consider, and it's a little bit out of order from the syllabus I gave you. We're taking reovirus next, and then the retroviruses will be after reovirus. So reovirus is unique in that the genome is double-stranded RNA. Reoviruses are the discovery of naturally occurring double-stranded RNA, and it was found that the genome is constituted by duplexed RNA. And so the family of viruses, Reoviridae. Viridae virus family. What Reo stands for is respiratory, enteric, orphan. REO. The name comes, the viruses were identified, isolated and identified during the search for the causative agent of polio. And this was one of the viruses discovered, and it was studied because, in part, because of the unique nature of the genome, it's double-stranded RNA, but it turned out, and it was isolated from human, but it turned out that there was no disease associated with the virus, and that's why it was called orphan, no disease, but it was then a respiratory enteric virus. And so the main member of the family that's now known as Reoviridae, and our prototype virus will be called Reovirus. And so it's a little confusing because the name of the group is the same as the name of the family. But subsequently, other viruses in this family have been identified that are important disease agents. But the prototype Reovirus that actually is now known as an ortho reovirus does not cause disease. Okay, so what are these viruses in molecular terms? So schematics of RNA viruses. And on your handout you have the cell for the replication of reovirus. So what are these viruses in the context of what will make our family? So the virions are icosahedral virions, and it's kind of behind the PowerPoint here, but in that summary schematic up in the corner. Icosahedral virions, 75 to 80 nanometers in size. They're naked, but they're a little unusual in that they have a double capsid shell. The genome of these viruses is RNA, but as I said, it's double stranded. It's the discovery of double-stranded RNA as a naturally occurring molecule. And furthermore, the genome is segmented. And dependent upon the group of reovirus, there's either 10, 11, or 12 segments of double-stranded RNA. So the genome is segmented, and those segments fall into three sizes. So that's why there are kind of three different sizes of the duplex. The multiplication of this family of viruses occurs completely within the cytoplasm of the infected cell. So all of the events will occur in the cytoplasm of the infected cell. Nuclear functions are not required. And you might get in the order of 500 PFU per cell produced out in terms of infectious particles from one virus particle infecting the cell. In terms of the rate at which this happens, the multiplication rate is a bit slower than measles and flu virus. And measles and flu virus, our myxoviruses, were a bit slower than polio and rhabdoviruses, which were the quickest of the viruses that we've seen in terms of the relatively short period of time required for a single cycle replication. For reovirus, it's in the order of 15 to 20 hours that are required from the time we infect to produce a plateau of progeny variants. So it's slower than the other viruses. All right, what are the groups of these viruses, and how has this developed since the initial discovery of the virus that's now known as an orthoreovirus? So the main groups of our viruses are, firstly, the original one is now orthoreovirus. So orthoreoviruses, and orthoreoviruses, in terms of the number of segments, these viruses have 10 segments of double-stranded RNA. And they're found in mammals. Again, really very important in working out mechanisms of pathogenesis in the mouse model. They're also found in birds. And in their natural host, no disease associated with. That goes back to the O and the original discovery of orphan. Then the second group of reoviruses, of the newer members, are what's known as the rotaviruses. And rotaviruses are very important pathogens. There are 7 subgroups of rotaviruses that are A through G. And these viruses rotaviruses have 11 different segments of double-stranded RNA that constitute the genome. And as I said, they're important pathogens, both in the human, in infants. They're a major cause of non-bacterial gastroenteritis. Non-bacterial, i.e. diarrhea. And globally, it's a major cause of death among newborns when infected with the rotavirus. It's not limited to humans. It's the same situation in cattle, bovine rotaviruses. Newborn calves, where they're very often in close quarters, the virus is effectively transmitted. Major cause of gastroenteritis and death. And we'll be finished in just a second here. So the third group of Reoviridae that I want you to know about are the orbiviruses. These likewise include some important pathogens. These viruses have 10 segments and important viruses of sheep, disease in sheep. The other viruses that are Reoviridae include coltiviruses, and the coltiviruses have 12 segments, and you find these in arthropods. And then there are phytoreoviruses in plants, and there are aquareoviruses in fish. And these either have 12 or 11 segments. They will not be considered relevant. Our prototype will be orthoreovirus, which we'll discuss on Friday. Have a good day. So Friday, I hope to finish reovirus and we'll touch upon rotavirus, of course,